You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is October 20th, 2025. We will be continuing our basic immunology course discussing Sompayrac chapters 13 through 17. This is presented by Dr. Nikita Raje, an associate professor of internal medicine, pediatrics, and is the section chief of allergy and immunology at Children's Mercy Hospital in Kansas City.
So the learning objectives for chapter 13 to 17 is going over immunodeficiencies, vaccines, immune system in case of cancers, immunotherapy, and then COVID-19 and the immune system. So again, just a slide for the immunodeficiencies. We'll talk about these in throughout the year, but this was what's included in the Sompayrac, how different parts of the immune system can get affected, as in the communication between T and B cells. If it's affected with CD40 ligand defect, it will cause hyper IgM. Absence of T and B cells will lead to SCID. Complement defects in neutrophils. There can be Treg defects that lead to, say, for example, IPEX and IPEX-like disorders. Absence of mature B cells, like in AGAM, or B cell defects. Other B cell defects give rise to antibody deficiencies, toll-like receptor defects. There can be defect in IL-1 receptors, and then there are bone marrow failure defects. So just a list here. I wanted to point out that the Sompayrac and Abbas actually go over secondary immunodeficiencies, including HIV/AIDS. I don't want to go over a lot in detail, but just kind of knowing that how that progression of the natural course of HIV infection occurs, where in the acute phase, if you look at the viral load, it goes up pretty quickly, the yellow line, right? And then as it moves through the acute phase, the viral load kind of starts going down, but it just continues to stay there at a lower level in the chronic phase. In the initial part, the virus-specific CTLs go up and continue to stay up. However, through that chronic phase, but when there is profound, over time, this HIV infection can cause profound immunosuppression, where the total number of helper T cells go down, and in that stage of AIDS is where the CD4 cells go down. The virus again starts going up, and the CTLs will also decline in number. So that's just the normal progression. There are really good anti-HIV medications, antiretroviral medications at this point that can control this natural progression. Moving on to talking about vaccines, there are the...
Dr. Raje, I don't think your slides are shared, just heads up.
Thank you for pointing that out.
Just when you transitioned a second ago.
Let's see, where is it, where I can share again. Thank you for pointing that out. All right, you see it?
Yes.
All right. So that's what I was showing, the figure where the natural progression of HIV/AIDS occurs and how the numbers go down with natural progression, and then the viral load goes up again. All right, vaccines. There are inactivated vaccines and live attenuated vaccines. Both types of vaccines give rise to memory helper T cells and B cell response. It is only the attenuated live vaccines that give rise to CTL response. So there are different kinds of inactivated vaccines, such as the whole virus is killed and there are preservatives that are added, like formaldehyde, and those are used as vaccine. Or the toxoid is purified and treated with aluminum salts and given as vaccines in case of diphtheria or tetanus. Acellular, which means only the part of the cell is used, such as in pertussis. There could be a subunit, which means part of that viral protein is produced by genetic engineering, such as in hepatitis B or HPV vaccine. Then there are carrier vaccines, which means the gene is introduced in the non-disease causing virus and introduced like a Trojan horse, like Ebola vaccine. And then the RNA, mRNA vaccine, as in COVID-19 vaccine. Live vaccines are MMR, varicella. Those are the live viral vaccines. Yellow fever is another one. These cause herd immunity, but unfortunately can cause infection in immunosuppressed, or if there is an even in an immunocompetent host, if there is a mutation causing disease. So how do these vaccines work? So vaccines have to be able to give those two signals that we need for activation of the immune system. The first signal and the second signal, right? So the first signal is the foreign signal, which is provided by the microbial protein. But the second signal has to activate the innate immune system, showing that there is a danger. And that's provided by the adjuvant that's added if it is not a whole killed vaccine. And that could be the LM that can be used or modified LPS that can be used to provide that danger signal that is used in multiple vaccines. All right, so why is our immune system not able to protect against cancer in some cases? And the reason is there is a balance between these genes called proto-oncogenes or tumor suppressor genes. And because of that, the defect in either the suppressor genes or activation of proto-oncogenes, there are various types of cancer that can occur, thinking of carcinomas versus sarcomas, or the solid organ tumors, or the blood cancers like leukemias and lymphomas. And either these can occur spontaneously or they are because of a virus associated defect that causes the cancer. Typically, the immune system that helps in surveillance of these cancer cells includes CTLs, cytotoxic T cells, or the natural killer cells and macrophages that are trying to look for any damaged cells that they can clear out. All right, we talk about immunotherapy, which is on the rise now, that is being used for various different disorders. So how is this immunotherapy produced and given? So the concept came around years back with this concept of hybridomas, where the cancerous B cell line or the immortal cells, right, were fused with cells that plasma B cells that make the antibody of choice. And these cells are fused, leading to what is known as hybridoma, and it is an immortal antibody cell line that continues to produce the antibodies with the same specificity that can be used to be treating that specific antigen. And then there was this concept that came around called CAR T cells. So these CAR T cells are cells that have a external part that is a single chain antibody that can bind to the target antigen that is needed to be targeted. And that antibody molecule is bound or fused to a co-stimulator and a signaling molecule, which is a CD3 zeta chain. So CD28 is used as the co-stimulator. So the first signal interacts with that antibody. You get the second signal because of CD28. And then once that activation occurs, you need a signaling molecule, which is the CD3 zeta chain that's used for providing that signal that can lead to activation of the immune system. So these are called CAR T cells. All right, there you go. Last slide, COVID-19. We know SARS-CoV-2 is one of the largest RNA virus. It has its single-stranded RNA virus that has 30,000 bases. It infects the ACE2 receptor, And the COVID-19 vaccine will activate the immune system where it will interact with TLRs such as 378, will lead to type 1 interferon response. There are these interstitial macrophages and alveolar macrophages that the virus might come across that will help in fighting that infection. And then there is a response from Th cells, as well as antibody response with IgG and IgA. So all that in a nutshell. And that's all I had to share today.